warm good morning to all of you and welcome to Unacademy. This is Surbhi, your educator, and welcome you all in today's class where we are going to discuss about the cell cycle regulation. तो आज हम सेल साइकिल रेगुलेशन के बारे में बात करने वाले हैं. And before starting the session, let me confirm once whether I am audible and visible or not. Then start करेंगे. Okay, so let's start with our session now. So in today's session, we are going to discuss cell cycle and its regulation. So basically, first of all, I would like to tell you few benefits of Unacademy, why you should join Unacademy. Okay? Unacademy के ऊपर आपको मिलता है daily live classes, live test series and quiz sessions for your performance evaluation, structured courses for your exam, and unlimited access to all the content. तो ये 4 बेनिफिट्स अनअकैडमी आपको देता है थ्रू इट्स प्लस सब्सक्रिप्शन. अब आप प्लस सब्सक्रिप्शन लेते हो एंड अपार्ट फ्रॉम प्लस सब्सक्रिप्शन अनअकैडमी के प्लेटफॉर्म पे आपको फ्री क्लासेस भी मिलती हैं एंड दीज फ्री क्लासेस आर योर स्पेशल क्लासेस. सो इफ एनी स्टूडेंट हु हैज नॉट येट जॉइन अनअकैडमी, तो अगर आप अनअकैडमी ज्वाइन करना चाहते हो तो कैसे कर सकते हो? उसके लिए आप अनअकैडमी लर्निंग ऐप डाउनलोड कर लो फ्रॉम प्ले स्टोर. आपको पहले फ्री क्लासेस देखनी है, मानो आप पहले प्लस सब्सक्रिप्शन नहीं लेना चाहते, तो यू कैन इजीली गो फॉर स्पेशल क्लासेस एंड इन दीज स्पेशल क्लासेस यू कैन वॉच ऑल द स्पेशल and the special class videos just by using my code Surbhi01 to unlock all the special classes. एंड जब स्पेशल क्लासेस अनलॉक हो जाएगी तो आप सारी क्लासेस देख सकते हो, जैसे कि मेरी डिफरेंट क्लासेस वहाँ पे अवेलेबल हैं और आप सारी क्लासेस देख सकते हो बिल्कुल फ्री. एंड इफ यू आर इंटरेस्टेड टू जॉइन प्लस, so for Unacademy Plus subscription, you can either go for a 12-month or a 6-month plan or a 24-month plan. And जो plan भी आप परचेज करना चाहते हो, you can use my code Surbhi01 to get 10% off on the overall subscription. ठीक है students? And if you are interested in another thing, that is the iconic subscription. So Unacademy के ऊपर iconic subscription है CSIR की category में, and in this category, in this subscription mode, you will get extra benefits like आपको यहाँ आपका personal coach मिलेगा, study planner मिलेगा, बूस्टर सेशंस मिलेंगे, डेडिकेटेड डाउट क्लियरिंग सेशन मिलेगा, प्रिपरेटरी मटेरियल दिया जाएगा, अनलिमिटेड प्रैक्टिस, एसओपी प्रिपरेशन, स्ट्रक्चर्ड कोर्सेज, ये सारी चीजें आपको अनअकैडमी के आइकॉनिक सब्सक्रिप्शन पे मिलेंगी. तो बच्चे की आपकी चॉइस है कि आप प्लस सब्सक्रिप्शन के लिए जाना चाहते हो अनअकैडमी पे, या फिर आप आइकॉनिक सब्सक्रिप्शन के लिए जाना चाहते हो अनअकैडमी पे, या फिर आप हमारी स्पेशल क्लासेस देखकर प्रिपरेशन कर सकते हो. और इसके साथ साथ बहुत सारे टेस्ट सीरीज भी हैं बच्चे, तो आप टेस्ट सीरीज भी देख सकते हो और टेस्ट सीरीज भी ज्वाइन कर सकते हो. तो अनअकैडमी आपको बहुत सारी डाइवर्स्ड ब्रांचेस के थ्रू प्लेटफॉर्म प्रोवाइड करता है. अब आपकी ये रिस्पॉन्सिबिलिटी है कि आप उस एग्जैक्ट प्लेटफॉर्म को चूज करो जो आपको चाहिए और अपनी प्रिपरेशन करो, एग्जाम क्लियर करो, बिकॉज मेन फोकस इज टू क्लियर योर एग्जाम. एंड announcements now, coming to new batches announcement. So Evolve CSIR December 2021, a new batch for physical sciences is going to start on 17th March. From yesterday start हो चुका है. Now Evolve CSIR UGC December 2020 batch for chemical sciences, नई batch start हो चुकी है. And another thing is Unacademy Accelerate program, that is a scholarship test. So this Unacademy Accelerate program for mathematical sciences ये होने वाली है 21st March को, संडे को. So those student who are related to mathematical backgrounds, और अगर आपके कोई दोस्त है तो उनको आप बोल सकते हो to give this exam of Unacademy Accelerate to gain maximum scholarship on Unacademy Plus subscription. Okay, so now we should start our lecture. चलिए स्टार्ट करते हैं। हम आज बात करने वाले हैं सेल साइकिल और उसके रेगुलेशन के बारे में। तो द रेगुलेशन ऑफ सेल साइकिल, तो पहले सेल साइकिल को डिस्कस करें। तो सेल साइकिल कैन बी डिवाइडेड इनटू टू मेजर फेजेस बेस्ड अपॉन द सेलुलर एक्टिविटीज दैट आर विजिबल अंडर अ लाइट माइक्रोस्कोप. What is the M phase or mitotic phase? The process of mitosis during which the duplicated chromosomes are separated into two nuclei is known as M phase, और इसको हम कैरियोकाइनेसिस भी बोलते हैं. मीन्स इट इज द फेज जहाँ पे क्रोमोसोम सेपरेट होते हैं, न्यूक्लियस सेपरेट होता है. ठीक है. तो एम फेज और माइटोटिक फेज और कैरियोकाइनेसिस इंक्लूड द प्रोसेसेस ड्यूरिंग विच the chromosomes are separated and two nuclei will be formed. So this is the M phase. Another phase is cytokinesis phase, and during this phase the entire cell divides into two daughter cells. So here the entire cell, जहाँ पे nucleus already separate हो चुका है, so this cell will completely divide into two daughter cells, and this phase is known as cytoplasmic division or cytokinesis, where we will get exactly two cells. In karyokinesis, the cell is only one, but the nucleus becomes two. Okay. But in this case of cytokinesis, two daughter cells will be formed. But in the M phase or mitotic phase, cell will remain one, but two nuclei will be formed. Okay. Good morning, Deepanvita. It's okay. Fine. Good morning. सो एम फेज के बारे में हो गया कि न्यूक्लियस डिवीजन होता है बट सेल विल रिमेन ओनली वन. बट इन साइटोकाइनेसिस टू सेल्स विल बी फॉर्म्ड विथ टू न्यूक्लियस. एंड देयर इज अ इंटरफेज आल्सो, एंड इंटरफेज इज द पीरियड बिटवीन सेल डिवीजन. मींस एक माइटोटिक डिवीजन कंप्लीट हुआ और दूसरा माइटोटिक डिवीजन स्टार्ट होने के बीच में जो टाइम पीरियड होता है दैट इज नोन एज इंटरफेज. एंड इन दिस फेज द सेल विल ग्रो, the cell will make proteins, the cell will make enzymes, the cell will replicate its DNA. मतलब कि finally the cell will prepare itself for the another round of division in the interphase. Okay, and interphase have three subphases, G1s and G2. We have already discussed. So talking about M phase, so M phase usually lasts only an hour. Okay, so mammalian cell usually invest its most of the time in the interphase, and the period of interphase also vary. So interphase may vary up to weeks or days or even a year for different type of cell and for different type of conditions. So unfavorable conditions will give you more longer interphase. Favorable conditions, enriched media will give you more less interphase. Okay, so this is about the cell cycle and its major points. Now we will move to our regulation part. So अगर हम कंट्रोल ऑफ सेल साइकिल की बात करें तो देखो सेल साइकिल हो रहा है, इंटरफेज होता है, सेल ग्रो कर जाता है. ठीक है, ग्रो करने के बाद वो एम फेज में एंटर करता है. एम फेज हो गया, साइटोकाइनेसिस हो गया. तो ये प्रोसेस क्या हमेशा चलती रहती है? ऐसा तो नहीं होता होगा ना कहीं तो चेक किया जाता होगा कि इंटरफेज में सेल प्रॉपर्ली ग्रो हुए हैं या नहीं हुए हैं. देन इन द एम एस फेज डीएनए कंप्लीटली रेप्लिकेट हुआ है या नहीं हुआ है. इन द एम फेज न्यूक्लियस क्रोमोसोम्स प्रॉपर्ली सेपरेट हुए हैं या नहीं हुए हैं. तो इन ऑल दीज फेजेस, इंटरफेज, एम फेज, देयर आर लॉट ऑफ चेक पॉइंट्स. ठीक है, और कंट्रोल पॉइंट्स. सो इनिशियली दीज कंट्रोल पॉइंट्स एंड चेक पॉइंट्स आर नॉट डिस्कवर्ड, बट इन 1970 only it was discovered that cytoplasm of cells contains certain regulatory factors that will affect the cell cycle activity. Before 1970 there was no such factor discovered जिसको बोल सकते हैं कि वो सेल साइकिल को रेगुलेट करता है. ठीक है, but it was in 1970 जब ये डिस्कवर किया गया that there are certain factors present in cell cytoplasm that decides the activity of a cell cycle. सेल साइकिल होगा नहीं होगा, ये सारी चीजों को डिसाइड करने के लिए देर आर फैक्टर्स प्रेजेंट इन द साइटोप्लाज्म. एंड दीज काइंड ऑफ फैक्टर्स, हाउ दे केम टू नो? सो दे परफॉर्म्ड फ्यूजन एक्सपेरिमेंट. सो फ्यूजन ऑफ मैमल सेल्स दैट वेयर इन डिफरेंट स्टेजेस ऑफ द सेल साइकिल. सो डिफरेंट सेल के फ्यूजन के बाद दे केम टू नो दैट there are certain factors. So in one experiment, they fused mitotic cells with cells in other stages of the cell cycle. एक माइटोटिक फेज का सेल है, इसको दूसरे फेज के सेल के साथ मिक्स कर दिया. नाउ द माइटोटिक सेल हैज सर्टेन फैक्टर्स इन इट. माइटोटिक फेज के अंदर कुछ फैक्टर्स होंगे, कुछ प्रोटीन्स होंगे, एंड दीज प्रोटीन्स विल इंड्यूस द अदर सेल टू ऑल्सो गो इनटू द माइटोटिक फेज. So this is what happened when they performed the fusion experiment. First of all, एक M phase के सेल को अगर लोगों ने इंटरफेज के सेल के साथ क्या किया, साइंटिस्ट ने फ्यूज कर दिया और ये देखा कि सारी की सारी सेल जो है उनके अंदर क्रोमेटिन कंडेंसेशन स्टार्ट हो गया. मींस क्रोमेटिन कंडेंसेशन इज अ फीचर ऑफ प्रोफेज. है ना स्टूडेंट्स, क्रोमेटिन कंडेंसेशन किस फेज का फीचर है, प्रोफेज का. तो जब एम फेज और इंटरफेज को फ्यूज कर दिया तो एम फेज के न्यूक्लियस एज वेल एज इंटरफेज के न्यूक्लियस में क्रोमोसोमल कंडेंसेशन स्टार्ट हो गया. बट इंटरफेज के सेल्स तो अभी इंटरफेज में है, वो कैसे कर सकते हैं क्रोमोसोमल कंडेंसेशन? बिकॉज एम फेज के सेल से कुछ फैक्टर्स, कुछ एंजाइम्स, कुछ प्रोटीन, एनीथिंग, दे may transfer themselves to the interphase cell and they will induce these cells to undergo chromosomal condensation. That means mitotic phase cells के पास कुछ ऐसे factors होते हैं उसके cytoplasm में जो कि other cells को भी stimulate कर सकते हैं to undergo M phase. Okay? So when you fuse a cell having the G1 stage and a cell having the M phase, तो G1 फेज के सेल में भी M फेज के कैरेक्टरिस्टिक दिखने लगेंगे. दैट मीन्स द G1 फेज सेल विल स्टार्ट टू हैव क्रोमोसोमल कंडेंसेशन, प्रीमैच्योर क्रोमोसोमल कंडेंसेशन. इज इट क्लियर टू ऑल? कंट्रोल ऑफ सेल साइकिल डिस्कवर्ड इन 1970 बाय फ्यूजन एक्सपेरिमेंट. सो व्हेन M cell is fused with G1 cells, then the factors present in the M cell will drive the G1 cell to also enter into the mitotic phase that will result in the chromosomal compaction. Is it clear, students? Clear? Okay. Okay, बच्चों ठीक है. So next is if a mitotic cell was fused with a cell in the S phase. Okay? एक माइटोटिक फेज के सेल को अगर आप S फेज के सेल के साथ फ्यूज कर दे तब क्या होगा? So the S phase cell chromatin also began to become compact. ठीक है, तो S phase के सेल्स भी क्या होने लगेंगे, S phase के क्रोमोसोम्स का भी कंडेंसेशन होने लगेगा, कॉम्पैक्शन होने लगेगा. लेकिन however we know that S phase के सेल्स क्या होते हैं, S phase के सेल्स में रेप्लिकेशन हो रहा होता है क्रोमोसोम्स का, so they are kind of relaxed, है ना, मीन्स वो ओपन रहते हैं, अनवाइंड रहते हैं, so they are kind of relaxed and loose kind of structures. So replicating DNA is very sensitive to damaged, and because when you fuse a M cell with S phase cell अपने replication को छोड़ के क्रोमोसोम का कंडेंसेशन करवाने लगेगा, and its chromosomes are generally very sensitive during the replication because वो unwind हो चुका होता है. So in this phase, when the condensation of S phase begin, then this will lead into the generation of chromosomal fragments. Okay? Because chromosomes जो होते हैं यहाँ पे बहुत weak होते हैं, unwinds होते हैं, so when you fuse M cell plus S cell, then S phase chromosomes will also become condensed, and during condensation these chromosomes will form fragments. Okay, these chromosomes will form fragments. So S phase के साथ fuse करने पे you will get chromosomal condensation, but chromosome will form fragments upon condensation. So the result of these experiments suggested that cytoplasm of a mitotic cell contained diffusible factors, means factors that can move from one cell to another cell, and these factors can induce mitosis in a non-mitotic cell. But let's assume a situation when you fuse a G2 cell with a G1 cell. So in this case, G1 cell will have factors, G2 cell will have factors, but G2 cell cannot enter into the G1 phase because this is the reverse. And G1 phase also cannot enter into the G2 phase because in between G1 and G2 there is a S phase required. Okay. So here these type of complications were also there, but scientists ने initial time पे ये discover किया पहली बार 1970 में that there are certain factors present in the cytoplasm, and उन्होंने उसको बोला diffusible factors, and these factors will be responsible for controlling the cell cycle. Okay, clear? Clear, students? हाँ जी बच्चे, clear? One second. Okay. So next is your type of regulations पे आते हैं. तो ये factors होते क्या है, ठीक है? So basically बाद में ये discover किया गया that entry of a cell into the M phase is initiated by a protein called as maturation promoting factor or mitosis promoted factor, that is MPF. Okay, so it was discovered that the induction of a cell into the M phase is basically initiated by a protein that is known as maturation promoting factor. This maturation promoting factor consists of two subunits. A subunit with a kinase activity that transfers a phosphate group from ATP to a specific serine and threonine residue of the protein substrate. So the first type of subunit is the subunit that has a kinase activity, and kinase activity, it will transfer a phosphate group from ATP to specific serine molecule and threonine molecule on the protein substrate. The first molecule is a kinase. First is an enzyme that has kinase activity, and next is a regulatory subunit known as cyclin. So this maturation promoting factor is a complex protein composed of two subunits, a catalytic subunit that has kinase activity and a regulatory subunit known as cyclin. Okay. So when the cyclin concentration is low, when there is no cyclin, then the kinase lacks cyclin subunit and as a result it is inactive. So let's say this is kinase and this is the binding site for cyclins. So when cyclins are not there, then this kinase will remain inactive. When the concentration of cyclin increases, when cyclins are there, cyclin will go and bind to the regulatory site, and this causes the kinases to become active, and these kinases will drive the cell to enter into the M phase. Okay. So when the concentration of kinase or cyclins is low, then the kinases are inactive. But when the cyclins concentration increases, then they cause the kinases to get active, and these kinases will allow the cells to enter into the M phase. So this is about the role of kinase. So there is a kinase, there is a regulatory protein cyclin, and this common structure is known as MPF, that is maturation promoting factor. And later on, these maturation promoting factor like enzymes are known as cyclin-dependent kinases. Okay, because they are what? Kinases, and their activity will depend upon the level of cyclins. अगर cyclins ज़्यादा है तो kinases will be activated, अगर cyclins कम है तो kinases will be deactivated. So kinases whose activity is dependent on the level of cyclins. So they are cyclin dependent kinases, और इसको short form में हम क्या लिखते हैं? CDK, cyclin dependent kinases. Okay, clear? अच्छी students, clear? चलिए चलिए फिर आगे बढ़ते हैं नेक्स्ट स्लाइड के ऊपर. That is about how the cyclins will regulate. Okay. So first of all, whenever a cell is in the G1 phase, then there exists a G1 cyclin. So the first transition point occurs in the G1 phase, late G1. And in this phase, the cell decides whether G1 phase is completed, the cell is completely grown or not, the cell size is enough for division or not. So basically, in the G1 phase, there is a cyclin that decides whether a cell should divide or not divide. Okay. Then after that, another checkpoint is at the end of the G1 phase. Okay. So that is known as G1s Transition Checkpoint. And in this G1s Transition Checkpoint, the cell will check if all the factors are available or not for DNA replication, if all the proteins, ATP, enzymes required for the DNA replication is present or not. And this checkpoint is known as G1s Transition Checkpoint, and it required G1s cyclins. So if there is a G1 checkpoint, so it required G1 cyclin. If there is a G1s Transition Checkpoint, then it required G1s cyclins. Then once this transition happens, then the S phase will start. And at the end of S phase, there is another checkpoint, that is S phase checkpoint, that require S phase cyclins or S cyclins. So at the end of S phase, there is a checkpoint that check कि डीएनए का रेप्लिकेशन कंप्लीटली हुआ है कि नहीं, इज देयर एनी डैमेज, इज देयर एनी मिसमैच द both daughter DNAs. So all these things will happen, and finally S cyclins will allow the cell to enter or to pass on the S phase, complete the S phase, and finally comes the mitotic phase. Okay, or from S phase it will enter into the G2 phase, and then to transit the cell from G2 to M, another checkpoint is there, that is G2M checkpoint or M checkpoint, and this M checkpoint again check whether all the components are there for the mitotic phase initiation or not. So basically there are different cyclins and transition points of the cell cycle. Basically there are approximately, अगर हम बात करें, so the cell cycle okay, so cell cycle transition points basically कितने हो गए, 4. First is the G1 checkpoint that requires G1 cyclin. Next is the G1s transition checkpoint that requires G1s cyclin. Next is S cyclin, and next one is the M cyclin. Okay, so if there is G1 cyclin present, so this G1 cyclin will go and bind to the CDK kinases, and CDK kinases here are CDK2. So if S cyclin or G1S cyclin is there, they will go and bind to CDK2, and CDK2 will be activated, and CDK2 will phosphorylate proteins, enzymes that are required for transition from G1 phase to S phase. So S phase will start with the help of CDK2 G1S cyclin, and now these CDK2 will also initiate the synthesis of other cyclins for S phase or for M phase. So when S phase continues, then S phase start हो जाएगा, DNA replication होगा, and at the end another checkpoint is there, that is S phase checkpoint, that depends on the level of S cyclins. So S cyclin check करेंगे CDK को activate करके. CDK will activate different enzymes like DNA repair enzyme to check whether any DNA damage is there or not. And when there is no damage, then the cell will enter into the G2 phase, and G2 will undergo another checkpoint, that is the M checkpoint that requires mitotic cyclins. So this is how the whole process of DNA replication followed by cell replication is being achieved. Is it clear? The whole story? And students, is it clear? The whole story, the 4 checkpoints and the 4 cyclins. Now coming to this 4 cyclins, तो ये 4 cyclins कौन से होते हैं? So I tell you. So see this image. So these are the 4 cyclins. ठीक है? So different cyclin CDK complexes will target different cell states. For example, G1 सेल जब होगा तो G1 सेल में जो G1 चेकपॉइंट होता है, तो G1 चेकपॉइंट को क्रॉस करने के लिए the cell require G1 cyclin, and G1 cyclin is cyclins D. Cyclins D are the G1 cyclins. So cyclins D make a complex with CDK4. Okay, cyclin D, CDK4 or CDK6 will make a complex. CDK will get activated. Now this CDK will go and phosphorylate different proteins required for S phase or required for transition into the S phase. Okay, so the first cyclins to be activated is cyclin D, and this cyclin will activate CDK4 or 6. Now once G1 phase happens, then the next checkpoint is at the G1S transition. Now the G1S transition is taken care by the G1S cyclin, and G1s cyclin here is known as cyclin E. So E cyclin will make a complex with CDK2, and these two complexes will allow the cell to transit into the S phase. Now completion of S phase is marked by another cyclin, that is S cyclin, that is known as cyclin A. So cyclin A will make a complex with CDK2, and this kinase will now allow the cell to complete its S phase and to move into the G2 phase. And finally at the G2 phase, there is another checkpoint at the end that allows the cell to transit into the mitotic phase. So it is G2M transition checkpoint that require M cyclin, and M cyclin here is cyclin B or A, and cyclin B or A will make a complex with CDK1. So here there are four cyclins: cyclin D, E, A and B. Cyclin D make a complex with CDK4 or 6. Cyclin E make a complex with CDK2, cyclin A make a complex with CDK2, and cyclin B make a complex with CDK1. So this is the ultimate thing you need to remember, that what are all cyclins, and these cyclins will make a complex with which type of CDK molecule. Clear, students? Okay. So that's all for today's class. We'll end our class here now, and remaining thing we will discuss in our upcoming classes. That's all for today. And if you have any doubt, let me know in the comment section. Anybody has any doubt, let me know. And if no doubt, then जब आप recorded video देखो तब कोई doubt आए तो आप अपने doubt को comment में mention कर देना, ठीक है? And student, if you are new to this channel, तो channel subscribe कर लो. You can hit the bell icon to get the notification. And आप वीडियो को like और share भी कर सकते हो अगर आपको अच्छी लगी है तो, ठीक है? And if you want to join my classes on Unacademy, for that you have to download Unacademy learning app, create your profile, log in, and then you can watch all my special classes for free just by using my code Surbhi01 to unlock all the special classes. And if you want to go for plus, then also you can use my code to get 10% discount. So that's all for today's class. Thank you so much, students. We'll meet again tomorrow. Thank you so much. Keep studying and all the best.